Good evening. I'm pleased to welcome everyone to the IMWG conference series, ASH 2017. This is in Atlanta, Georgia. You see a beautiful picture there of Atlanta, Georgia. The only correction I would make is that it has been snowing and extremely cold with freezing temperatures, which is quite a challenge for some of the logistics of ASH. But nonetheless, we're all glad to be here to hear a lot of new information and try to make sense of all of this to come up with the best treatment recommendations for our patients. I'm very pleased this evening to welcome two speakers who have joined us in the past. Very, very pleased to have these return speakers. Joe Mikhael from Mayo Clinic, Arizona. Welcome, Joe.
Thank you, Brian. It's great to be here.
Thank you. And Paul Richardson from the Dana-Farber Cancer Institute in Boston. So welcome again, Paul.
Oh, it's a pleasure, Brian. It's lovely to be here.
Very pleased to have you both. So for tonight, the topics are broken up into 6 segments. And this year, it is interesting that in each of these different areas, there were several interesting and sometimes quite important abstracts, updates. And so the idea would be to take a look at those, give some perspective, and see how they might change our day-to-day practice. So priorities for early disease, frontline options, the role of autologous stem cell transplant, maintenance recommendations, early relapse management, and then, of course, as always, we are interested in what are the new therapies. So if we start with the priorities for early disease, this year there were in fact a lot of analyses looking at what are the indicators of early disease progression, and tremendous interest in that. And so I will only show you a little bit of data. There was one quite interesting data set from the Mayo, Arizona, from Joe's colleagues, and quite interesting work where the team there have been quite involved in looking at MYC translocations, which seem to be especially important in early disease progression. And as you can see here, patients who did have early MYC translocations really were at much, much higher risk of early disease progression, really a dramatic lowering of the curve there. Now, another completely different approach, which has also been presented by Dr. Bruno Paiva from Pamplona in Spain, is starting to look at low levels of disease in the peripheral blood. And using the Euroflow combined Spanish method, the NGF method, which is sensitive at 10 to the minus 6, they have evaluated the blood for clonal plasma cells. So these curves here are looking at the presence or absence of clonal plasma cells in the blood in patients who have MGUS. And you can see here, if there are no clonal plasma cells in the blood, those low-risk MGUS patients remain stable. In the Smoldering cases, if there are plasma cells in the blood or clonal plasma cells, then it really does increase the risk of early disease progression. Now, just to draw your attention, Sigurdur Kristensen from Iceland, he gave an educational presentation about his iStop MM project, which is one that's supported by the IMF Black Swan Research Initiative. It is, in fact, already the largest population-based study ever for the plasma cell disorders. The goal was to screen 148,000 Iceland population above the age of 40. And we were just in Iceland for the 1-year anniversary when we learned that 80,000 have already signed up, and with blood samples and testing ongoing for about half of those already. And so there will be a randomization to regular follow-up, some more intensive follow-up, and then an arm where intervention will be offered for those who meet the criteria for high-risk smoldering, for example. The diagnosis, the initial diagnosis is based on the presence of a monoclonal spike. One background element is that all of these patients have molecular sequencing data, which has been carried out by the DCODE facility in Reykjavik in Iceland. So a lot of information is going to come out of that, which will be certainly informative about the predictors of progression. So my question to you guys is, with this kind of information, some molecular information, of which there's a lot of different types, mutational load, various things that people have looked at, some cellular information with cells in the blood, which has also been looked at by several groups actually, and then population-based information. What do you foresee will be the best way to diagnose early disease? And so perhaps, Joe, you might want to start with this one.
Yeah, well, I mean, it's a great question. I think it's a really important question because you know, so many of our patients are so anxiously waiting for each visit. Has my disease progressed? Has my disease progressed? And we've always had to wait on fairly archaic information, just an artificial rise in the monoclonal protein, or perhaps sadly the development of Crab criteria.
Yes, some bone
where a
lesions or
patient
something.
comes in with bone pain or kidney damage or something like that. So I think the drive to find the answer is critical. I'm not sure. I'm encouraged that we actually are finding multiple things. You know, the MIC study that our group presented, I think, was very fascinating. Although it's a pretty small proportion of those who will have it. I mean, if I can't be a prophet, but if I were to predict it, it's kind of like saying, what are the measures that we would see that we're going to see a rise or a drop in the unemployment rate in the economy? We look at 5 or 6 or 10 different things, and I suspect that we'll add to our usual clinical questioning some kind of testing. The blood testing is the most attractive because it doesn't
would be a simpler
involve,
test.
Exactly, multiple bone marrows.
Right.
So I suspect that hopefully we can be better at that because there's still going to be such a significant group of patients who don't need intervention.
Right.
But it'd be nice to catch those patients before they're in trouble. As I often say, you know, if someone's running towards a cliff, I don't have to wait till they're falling off the cliff to know they're in trouble.
Right.
But if the cliff is miles and miles away and they're going for a leisurely walk, we don't need to intervene earlier. So I think it's going to help us determine how fast a disease is growing and how far they are from the cliff.
Right, right. So, Paul, what's your perspective about this?
I think Joe put it beautifully. I mean, I think the important point here is that, you know, we've got multiple parameters that we can follow. And I agree completely that on the one hand, we have sort of hard benchline clinical criteria that we can use to sort of anchor our decision making. But at the same time, these new tools provide us with an ability to be that more, hopefully, you know, have that much more foresight as to what might happen. And I completely agree with Joe that the essence of intervention is prevention, as it were, to further organ damage and or consequences thereon.
Certainly want to avoid that. That's the goal.
Exactly. And the whole point is that if we've got, as we now have with novel therapies, opportunities for therapeutic intervention that are so much better than they ever were, we've got the opportunity to actually employ or deploy these to help our patients. Now, in my group, we're very fortunate we have Irene Gabriel, my colleague, who's doing phenomenal work in precursor disease and MGUS and Smoldering myeloma. And what she's showing very nicely is that you can actually intervene earlier with relatively non-toxic, simple treatments that can actually result in clinical benefit, we hope, recognizing there's work to do. But I agree with you, Joe. I particularly like your work with the c-MYC issue because, in a sense, that's a biological parameter that tells us, you know, what we see as a sort of vanilla may not be. And clearly here, this is not vanilla, and we need to react to it. And I think that those kinds of observations are vital so that we can be that much more precise in what we do.
Right, right. So not quite there yet, but potential.
I couldn't agree more.
I think maybe just a final comment would be it emphasizes again, especially to so many listening today, that incredible need of doing that basic kind of work.
Yeah,
you know, we need
gotta
new
do
drugs for myeloma. We need to do the large clinical trials, but understanding the mechanisms that underpin this disease are going to be critical for us to intervene at the right time.
Yes.
Right, right. And so obviously we are very excited to have the opportunity to be involved in the Iceland project, where we will learn a lot by tracking these patients carefully.
Yeah, I think that's phenomenal, Brian, and I think your initiative there really should be applauded. It's really something quite unprecedented, and I think that to see participation from the Icelandic population to such a degree.
They're so committed.
What a remarkable country.
They're on Facebook and encouraging each other to participate.
It's great. I mean, my simple point is, you know, for people looking at, you know, why? Well, why a lot? Because essentially they're committed in various other areas to sort of a stitch in time saves 9. And it's great to see that kind of participation from whole groups of people. It's wonderful.
Right, right. So we're going to move from this to something that you just touched on with Irene. what would be the management right now?
Right.
There are some management possibilities, and so we were quite fortunate this year at ASH to see presented a couple of quite important abstracts. So I'm just going to take us through slides from two of those important presentations. And so the first one is from the Spanish team led by Mary Mateos from the University of Salamanca, the CESAR trial. So obviously a very aggressive protocol for the treatment of high-risk smoldering myeloma, starting off with carfilzomib, lenalidomide, and dex, high-dose melphalan autologous stem cell transplant, 2 cycles of consolidation, and then 2 years of lenalidomide dexamethasone maintenance. So obviously this is the aggressive approach, and fortunately has been really tolerated reasonably well. And thus far, as far as the presentation, there are 35 patients who have gone through to the maintenance phase. So what have been the results? And I've just gone to the key endpoints here, which are 74% greater than CR, but I would draw attention to the blue bar at the bottom here, where 62% MRD negative at 10 to the minus 6. So really a substantial impact with achievement of that MRD level. And so this translates at this point into really excellent PFS and overall survival curves. And so this is obviously very important data to see what can be accomplished with this kind of a strategy. And there is an upcoming ASCENT trial, almost identical to this, where the induction will be daratumumab KRD. And so that will be starting in the first of the year. However, how about a more gentle approach? And we had the opportunity to see that at this year's ASH. And so this was the use of daratumumab monotherapy in smoldering myeloma. And the Centaurus trial had 3 arms, with the bottom one here being just basically the induction type of arm, and then an intense and long and less intense use of daratumumab. So 3 arms to assess the impact on an ongoing basis, or in this case, just with this initial cycle. And so the differences were seen in the overall survival. You can see here 56, 54, and then obviously much less with the short intense, as you would predict.
In response rate.
This is the response. Excuse me.
Yes, no problem at all. Yeah, not at all.
Response rate.
Yeah.
Yes, thank you. Yes.
Yeah.
And so this is obviously quite encouraging, but I think that this is, for me at least, somewhat sobering, is that in those 3 arms, you have obviously ongoing response in the continued therapy, but a rapid drop-off here with just that initial induction, as you might expect. So my question to you, you guys, is what do you think about this? What do you favor in terms? Really, I'm interested in the way that we should be exploring management in this setting.
Yes.
How do you view the cure approach, and how do you view the control approach? And so perhaps, Paul, you can tell us first what you think about this.
Well, I think thank you, Brian, for a fantastic summary of 2 very divergent approaches to a critical question. And I personally am struck by the fact that if you look at the continuous therapy with daratumumab compared to the transplant experience with intensive therapy pre and post, the results are very similar in terms of PFS outcome. And unfortunately, in Mary V's trial, as the slide showed, there was 1 catastrophic stroke.
Yeah, 1 patient had a stroke.
And died. And we know that there's a vascular toxicity to carfilzomib, which is being better understood. And in fact, there's some lovely work at the meeting that has teased this out.
Right.
So I think we have to be very careful.
Have to be careful, obviously.
And I think what we have to say, because arguably in a smoldering population, 1 death is 1 too many. In any population, one could argue it is. But the fact is, in this population, it's particularly true. I think that beautiful work, and I applaud the Spanish colleagues for doing such a careful and ambitious approach with, as you say so nicely on the slide, Brian, attempted cure. But I think that I'm struck by the quality of Dr. Sorry, it was Craig Hofmeister who presented the Darrow paper. I'm struck by the quality of Craig's work. It really, and his colleagues, it really does show that with a non-toxic approach, because if you look at the grade 1, 2, and 3 toxicities,
yeah, I didn't show that, but
it's really remarkable. I mean, everything was just here.
Yes. Oh, very
and
nice.
unfortunately, you know, when you look at, and Mariví did a beautiful job for preparation and very clear. When you looked at the grade 1, 2, 3s, they were out here. So the point is that, you know, and there was no quality of life report from Mariví's presentation yet. That will be essential. I was also struck by another thing, the MRD negativity.
Yes.
You know, 60%. That's interesting. But what's also interesting is when you look at some of the lovely work, for example, from Shaji with Ixazomib RD up front. In the high-quality responses, the MRD negative rate is 80% without transplant. You know, that's this once-a-week Ixazomib study that Shaji updated at EHA. And in the same study set, you know, with the twice-a-week approach of Ixazomib with an all-oral approach, we see the same without transplant. So I think the question becomes, how much is enough? And I think I applaud the Spanish group for the curative intent.
Absolutely.
But I think one has to be very careful because, you know, are we achieving a functional equivalent with antibody alone or antibody immunomodulatory?
Absolutely,
and
absolutely.
a proteasome inhibitor that's just a pill once a week.
So the ASCENT trial actually will start with Dara KRD without the transplant. So we'll have the data with Dara KRD.
I think that's very important.
And then, so in that trial, it's going to be with and without the transplant, just to be aware. So how do you view this? Paul is expressing his concern about the intensity of this type of curative approach in the smoldering setting. But would you see any negatives to the simpler Dara approach?
Actually, I do. So I'll be a bit controversial.
Okay.
I'm not convinced by the Dara work either. Those response rates were much lower than we would ever want. And I guess my philosophical approach to it, Brian, Paul, is to say, the approach that we took, would we change the criteria of myeloma from crab to slim crab?
Right.
We're drawing that line. Well, I'm not sure. I didn't change it. I just named it. But that step was, I think, a very important step. And if I could be a little controversial for a moment, what I'd love to see is a day where we have MGUS here and myeloma here and no smoldering anymore. Where we better draw the line and say, when someone crosses this line, you need treatment. And that treatment is an evolving phenomenon. As Paul has said, we may or may not be doing transplants in the future. We may be including monoclonal antibodies up front. But I worry about putting a separate category in the middle that we kind of half-treat. I'm not as worried about these Dara patients in the short term. Daratumab is a great drug, very well tolerated.
But in
I
the
worry,
longer
are we going to
term,
breed resistance? Are we not
treating
people long term? So I'm almost in between these two, where I think one is too little,
and then,
you know, we're Goldilocks here, a little too hot, a little too
cold,
maybe
exactly.
just right in the middle.
So I think that
I think if we go from let's say slim crab to slimmer crab or slimmest crab, who knows? But I think if we better define those points where we know someone is right on the verge of truly having myeloma and intervening the way we would treat everybody is probably safer than setting two standards.
So another point which I think might help us with this, in the Iceland study, what we are doing is actually giving KRD to the smoldering patients and in a randomized fashion, KRD to the myeloma patients. So actually we will be able to answer this point.
By comparing those two groups.
So we'll have the two groups. Does it actually make that extra difference to intervene that earlier? But we're using the same therapy, and so it hopefully get some information on that.
Yes, I agree with Joe that I think we have to be careful about this definition. I think in the future, actually, I do believe also that we will have true MGUS with some perhaps a high-risk MGUS for which there may be appropriate immunological interventions that are warranted. But in fact, ultimately, high-risk smoldering becomes active disease that we need to treat.
Absolutely.
And I think that what I'm struck by in our own experience, because we have a variety of approaches, and we've been using IMiD-based approaches, combining IMiD with Ixazomib under one of the studies that Irene is leading. And you know, I'm very impressed, actually, with how well that's doing.
Good.
I'm also very impressed by how single-agent antibody is well tolerated over time. And I think you're right, Joe, that we certainly don't want to get into a situation of, you know, betwixt and between. But at the same time, these efforts are very important because they juxtapose to what we've seen from our Spanish colleagues to say, well, you know, how much is enough?
Exactly.
And I think that, you know, we have to remember what we've learned now, we are learning from the determination trial, which is the long-term consequences of high-dose melphalan.
Absolutely.
As our patients live 10, 15 years, the real spectre of secondary MDS AML is becoming more and more important.
We definitely need to be cognizant of that.
Exactly.
Yeah, so thank you for that. Yeah, so moving forward to what do we do right now for the frontline patient? I'm going to look at that or present that in the non-transplant setting where we had excellent presentation, as well as in the transplant setting. So if we start off in the non-transplant setting, again, and this is actually is quite remarkable, is that we have this exact same investigator, Mary V. Mateos, from...
She's busy.
She's a busy lady and had really another excellent presentation, I would think you would agree, the Alcyon trial, in which she took what is certainly the standard of care in the non-transplant setting in Europe, outside of the US, the VMP, and added daratumumab to that in a randomized setting with over 700 or so patients, a large study, and then with the ongoing therapy to see the difference. You'll notice on the top part of this chart that the VMP treatment does stop. And I believe the primary reason for that is that is the standard of care which is approved in Europe. And so for that reason, it is the comparator, okay? But I'm just mentioning that point.
Good point.
But the results, striking improvement with the addition of the daratumumab. Not really fully mature study yet, but you'll see the hazard ratio over here of 0.5, obviously excellent, with striking difference at the 2 time points so far, 12 and 18 months. And so we also see what is very good, this striking difference in the achievement of MRD. And I think that it's clear, as we'll see later from the follow-ups of, for example, the Pollock study, that the addition of daratumumab really does enhance the tendency or the ability to achieve this MRD level. In this case, it's a 10 to the minus 5 threshold. So the question becomes, obviously, I think that on the other side of the Atlantic, the DARA VMP will certainly be considered as a new standard of care. But on this side of the Atlantic, I've given you some options to consider here. And so maybe I'll come back to you, Joe, to start with this topic. How do you view this?
Yeah. Well, first of all, I view it with tremendous enthusiasm. I mean,
yes.
we know that monoclonal antibodies are a huge part of our care of patients in myeloma. Up until now has really been in the relapse setting. I think most of us believe that it's going to come to the upfront setting.
Absolutely.
There's some challenges because our response rates from VRD and KRD are so good.
Yes.
You know, you're already at 90%. You've already got an A+. So you're going to get an A++.
And many patients with significant remissions and survival already.
Correct. So there's a cost issue. There's thankfully not a huge toxicity issue because it's so well managed, but so well tolerated. But I would say that this study is not going to immediately affect us in the U.S. as it would in Europe. Yeah. But what it does is it shows us that it's a safe thing to do. It's a feasible thing to do.
Kind of a proof of principle of these.
Correct.
Yes.
And honestly, each of those, I could argue that based on the patient in front of me today who's not going for a transplant, there could be benefits to all of those. We had an excellent presentation here at ASH putting 4 huge trials together, looking at either a lenalidomide-based or a bortezomib-based regimen and transplant ineligible, that overall they were both in many ways equivalent, but in the highest risk patients, we should consider ensuring we have bortezomib, and then in patients who are very elderly or very frail, we should consider lenalidomide. So there's a place for both drugs. There's a place for both drugs together, or potentially
or maybe VRD light, huh?
Correct. And if I tend to use VRD light in most of our transplant ineligible patients. Obviously, the studies are ongoing, but I think we will see daratumumab come up front, and there are going to be some patients who will experience very little additional toxicity and more benefit. But I'd also suggest that we'll see Dara-RD on its own. We might see Dara-VD on its own, and a very small subset of patients, maybe even Dara alone. So, so we have to look at, myeloma is multiple for lots of reasons.
Absolutely.
But I'm very encouraged by this study because I think it's going to mean more antibody use up front.
Yeah. So, Paul, what do you think?
Well, I think Marivy's had an amazing meeting and she does beautiful work. And in the European context, I agree that Dara VMP will be a new standard, and for a regulatory point of view will be critical. I think the VMP control arm, performed as I suppose we would expect it. I'm troubled a little by this, though, because I think as we move into the new randomized trial setting for our patients, first and foremost, control arms that are essentially applicable in only specific settings, and arguably, are somewhat, and I use this very carefully because I want to be careful how I say this. I'm not going to say VMP is obsolete, but it certainly isn't a wave of the future. And what I would say is that whilst it's performed very well, and clearly, having been part of the VISTA trial myself with Jesus and Marivy, I'm very pleased with the way it's performed in the past. Looking to the future, I think we have to think about control arms, because I think for our patients to have equipoise and to serve them well, we have to be thinking with our regulators, to our regulatory authorities, please don't box us with regimens.
No, we need to work with the regulators.
Exactly, to say, can we have control groups that really reflect modern practice?
And we almost never use melphalan up front in
Exactly.
transplanted eligible patients. I mean, that's a critical point. We've done surveys in the country, less than 2% now patients will receive melphalan.
And using the melphalan, you really are kind of closing that door, which can sometimes be an uncertain door to close, right? You want to keep it open.
A, you can do that, and B, of course, again, chronic melphalan exposure has with it its own challenges. So I think a couple of points to make there. I'm very excited by the data, because at the end of the day, what the team have done is established daratumumab as upfront. It will get approval on that basis, and to the very tough regulatory environment in the European setting, it will do what we need it to do. So good for patients in the long term, which is the key point. So I don't in any way want to diminish that, because that would be very, very unfair to this study. It's done a great job in doing it. I think the important point, though, is just what you've raised, Brian. What does it do to our use of the antibody in the upfront setting in other combinations? And I completely agree with Joe that in the non-transplant eligible population, the use of regimens like RVD Light, there was a beautiful presentation led by my colleague Betsy O'Donnell on RVD Light. We have a median progression-free survival with RVD Light in elderly population up to the age of 90 plus of 36 months. I mean, that's just gorgeous. And the thing is that beyond that, we also have the Exazimib data.
Which is great,
It is. So we're looking
the VRD actually in the SWOG trial in the elderly also have this kind of impressive benefit, as a matter of fact.
Exactly. So I think the point simply being that, and also at this meeting, there was a wonderful presentation by Peter Voorhees with the RVD combination plus Dara plus transplant, showing great safety going forward. So lots of exciting platforms with Dara.
So I'd like to move on so that we have a chance to cover the frontline setting. And so a few abstracts just to give us a little bit of information in this setting. So one that was interesting was from the French, the IFM trial, with an update focusing just at this meeting on the sensitivity at 10 to minus 6 for MRD. Obviously, patients negative at that level doing better. And the interesting thing that I'd like to hear your opinion about, though, is what about the impact on the treatment arm? And so these two curves at the top are for VRD and with or without transplant. And so obviously the MRD negativity is the driving piece of information here.
Identical.
And the additional piece of information is that it's those same MRD negative patients that have the better survival, irrespective of whether they had a transplant or not.
Exactly.
And so very, very interesting. The other piece of information I'd like to show you is, well, what about with carfilzomib, lenalidomide dex? We have a couple of pieces of data there showing that gives excellent results in the frontline setting. This is Jakubowiak's data. And then we have the corded data of the excellent results with carfilzomib lemdex. And this introduces the idea, is this possibly better enough to consider it here, which is actually part of the MSMART criteria. An interesting thing in the transplant setting was also the addition of daratumumab to the carfilzomib index. With also, this was largely a safety study led by Chari from New York, but also excellent response rates over the first cycles here, excellent deep responses, so safety, excellent responses. And so again, the question here is, how do you look at, with this information, the frontline choices? So maybe you want to start with this one, Paul.
Sure, I'm delighted to. I mean, obviously, I think the aggregate here is phenomenal. It's all very, very positive. I'm particularly impressed by the KRD data, and having partnered with Andre and at the time Todd Zimmerman before whilst he was in Chicago on the KRD transplant study, to see the very high quality of responses involved. And I think that these are building on the principle that the IMiD plus the proteasome inhibitor plus the steroid really matter, and are now a standard of care. Gives us, I think, a platform for real progress, and that clearly this reflects it.
Yes.
I think, however, what we have to be aware of, as you said so nicely, Joe, myeloma is multiple and in every way, and so tailoring therapy becomes now our priority. And I think Hervé's work is really important, because what it tells us is if you achieve MRD negativity, it doesn't matter if you have a transplant or not, you do just as well. So that allows us the capability to sort of step back and start to be more selective in our treatment options. So
do you agree with that? How are you viewing the role of the transplant now?
Yeah, I mean, I think it's fascinating work. It's important work. And if you told a patient, we can get you to the finish line with or without a transplant, they're going to say without a transplant, right? So we don't want to get patients to transplant. Now, of course, the study wasn't designed in that way. And I think as we look forward, so many studies are looking at making a decision based on MRD negativity as opposed to looking back. We're looking back now and saying, oh, these people did the best. And some of them had a transplant and some of them didn't have a transplant. But we didn't know who those people were at the start. And that's the problem. Not the problem, but that's the issue we want to determine. Because right now, I can't look at that and say, well, I'm not going to offer transplant to patients. We still see that in general, patients who had a transplant did better. But it does help us to appreciate the importance of MRD negativity. And of course, we have to wait to see in the long term what happens. I think what we'll probably end up seeing with time is, as we collect more and more of this kind of information, that we might pull back a little bit, paradoxically, from transplant, even though the study obviously endorsed it. Because we will start to do a little bit more, as opposed to saying you need this many cycles of treatment, then a transplant, then maintenance. We'll say, let's give you a shifting number of cycles and see what your depth of response is. And if you're not at the depth that we think you should be, maybe we'll go to transplant, or maybe we'll not have to go to transplant.
And in fact, Hervé proposed this, instead of just sticking with the 4 cycles, to consider like 6 cycles. Of course. Just without the transplant, to see what would be achieved.
And those 4, as came from 2 principles. One, in the early days when we used VAD, and we didn't want to give too much adriamycin for cardiac toxicity. And then we learned if we gave a lot more than 4 cycles of lenalidomide, it might compromise our ability to collect cells. So, but there's nothing biological about 4.
It's not a magic number.
6 could be better, 5 could be better, and in some patients 2 is enough. So again, we go back to this notion of looking at
being adaptive.
Exactly, adapt our therapy to the depth of someone's response.
And in the determination and the IFM work, we picked the 3 cycles of induction RVD based upon the fact we wanted to minimize neurotoxicity, especially across stem cell collection. And I think you're absolutely right, Joe, and I fully endorse Hervé's conclusion that this provides us with what we actually was part of the idea of the trial in the first place, which was to be adaptive and tailored. And that essentially what this information says is that we can use a tool to guide us in those decision-making processes, and we need to validate that tool, which is obviously so-called determination 2, the next generation of trials in this setting, where we're going to try to establish that. Because what I'm very struck by Hervé's data is this is without antibody. So when we add antibody to this platform, it will go to a next step in terms of being able to spare people
that much better. And we know, we were just referencing the fact that with the DARA, there could be a significant enhancement of that MRD achievement.
Exactly.
And so I'd like to take you forward just with this exact point. So at the meeting here, there were some data that influenced this as you move forward from the transplant into the maintenance setting with the assessment of MRD. So patients, some who have not achieved MRD, will achieve MRD as they move into a lenalidomide maintenance setting. And so I certainly thought this was quite interesting from the UK. This is a landmark study, which means that patients were studied after 6 months of lenalidomide maintenance and whether they had achieved MRD negative or not. And you can see obviously here, if they had not, then the outcome was much inferior to those patients who did move on with that extra improvement. So quite interesting. And so this leads on to, well, what are the prospective trials that we need to help evaluate in this sort of situation? And so this kind of structure from the Spanish with this upcoming trial, well, actually it's an ongoing trial, Activism RD, but then with the second randomization over here, which I think will be especially informative. I think something that we really need to look at to understand and give the best advice in the maintenance setting. And so a little bit looser question here about how do you approach maintenance. And so actually I know, Joe, you were part of the meta-analysis and wrote an editorial actually when the meta-analysis of lenalidomide maintenance came out this past year. So what are your comments about this?
Yeah, no, this is great work and it's advancing the field. I would say, to keep it simple, I would suggest that maintenance therapy is pretty well the standard of care now. We have the caveat that myeloma is multiple. There's still the occasional patient who, for their own desires and wills and based on the nature of their disease and certain very low-risk myelomas, it may be reasonable to not give any therapy and just monitor. But I would say in general we're giving maintenance, and then right now the best evidence we have is for lenalidomide maintenance.
Absolutely.
Where it may fall short a little bit, and I know I described this in my editorial, is in those highest risk patients, it appears that lenalidomide may not be enough by itself. And many of them we've demonstrated do benefit from a proteasome inhibitor. So the question is, should they go on bortezomib maintenance? Should they go on lenalidomide plus bortezomib maintenance? And can we maybe switch the bortezomib for ixazomib, being a pill and a little bit easier to tolerate? And I think those are the questions we still have yet to answer that we'll have information on. And then the last point is whether or not MRD negativity will influence it. That's why I really respect this design is brilliant. You know, this Spanish study, because it'll help us. We have this sort of intuitive thinking that if a patient becomes MRD negative, we can just stop all treatment. Well, that may not be the answer.
We need to.
They might need actually.
We need to figure that out.
I agree, Joe. I want to pick up on this because I actually think that, again, this has been an area of intense debate, and especially in the context of our French partnership, it was really a defining point in our work together because French regulators actually required us to stick to 1 year for the French trial. In reality in the US, we said, wait a second, our data tells us something quite different. So we actually have 1 year of maintenance in our French partners trial and maintenance until progression in our own determination study. And this is going to help understand that much better because the populations are identical. And we have 700 patients in the French trial and now 730 actually enrolled in the US trial. 720 will be randomized. The bottom line here is that will help answer it. Now, I have one question here on the Spanish design, which is, this is to me the challenge. MRD negative, do we stop?
Absolutely.
And I'm concerned about this because in our own French-American partnership, we've seen that in the MRD negative patients, and it depends somewhat on the tool you use, but nonetheless it's the observation that even in MRD negative patients at a median follow-up of 3 years, certainly by the flow technology, 20% relapse. And my own view on this is that if you're MRD negative, recognizing that it's a wonderful tool, but it may have some caveats, my own personal opinion is lenalidomide maintenance is your backbone and it holds. And then if you, to your point, Joe, where I fully agree with you, if you're MRD positive, what do you do that may be more?
Right, so I think it's kind of counterintuitive. It could be that for the MRD negative, you continue. And for the MRD positive,
you do more.
Absolutely.
Yeah.
Make some kind of a switch. And so I think that these are the things that we need to learn over time.
Yeah,
Yes.
no, absolutely. I mean, there's lots of ways of thinking about it. I try to think, describe to my patients, I think, it takes more gas to get up onto the highway.
Yes.
And once you're on the highway and you're cruising, you need less gas, but you still need some.
I couldn't.
Right.
And
There you
I
go.
wonder, I would love to be
Right.
able to stop treatment in patients, but if we can back it up a little bit at least and give them less and have them on drugs that give that continue their quality of life, that make it easier for them, but yet keep them away from disease recurrence. I think that's where we're going to have.
So I think
and
this
there are
point-
other studies that will look at
Yeah.
randomizing people to any treatment versus no treatment versus more treatment, as you've said.
Right, and I think that the whole sort of spectrum of this question changes as we have much more effective and better tolerated maintenance strategies. And
Where it can be more open-ended and safe.
well, quite. And Thanos presented some of the early data that's coming out with Exazimib-based maintenance, and it's extremely promising.
Absolutely.
It's very well tolerated, and it offers the option.
And very attractive for the patient.
Exactly, because
3
we
pills
saw
a month.
Right.
Right, and what was really interesting was to see the work from the MD Anderson group looking at ICSA plus R.
Right.
Very impressive. But the toxicity profile was impressive too, and not in a major way, but enough to say, ICSA alone, ICSA R alone. And my point is that we'll have choices for our patients.
Absolutely.
And whilst I personally think Len is a sort of backbone based upon the meta-analysis, and to which you may add things, for those patients who may not be tolerant of lenalidomide maintenance for a variety of reasons, ICSA alone as a standard may actually be a very useful alternative.
As we move forward, we might need to be flexible about what is a backbone,
Exactly right.
Well,
I agree.
In the meta-analysis,
That's very funny, Brian.
Right?
Speaking of someone with back pain, I can totally relate to that.
nearly a third of people had to come off maintenance.
Yes.
Exactly.
So it speaks
But
to the point that it's not one size fits all.
I couldn't agree more. And also, interesting enough, in Thanos' presentation of the collated ICSA experience, we didn't see that. The discontinuation rate was much lower.
Okay. I've got to pull you guys forward so that we can cover all these different interesting topics.
Yes, exactly.
So the next one is moving forward to say, well, what does happen if a patient has early relapse? And this has been, as you know, a very active area with many studies in the last few years. And so there have been some updates at this meeting. There's an update of the ASPIRE trial, which is obviously carfilzomib Rev-Dex versus Rev-Dex, with the improvement in overall survival to really kind of validate that benefit. And then update of the Pollux to sort of reaffirm this really amazing added benefit of daratumumab to Rev-Dex in the early relapse setting. And so really updates at the meeting to assess that. And so these are the little bits of extra information from ASH, and then we have all of the information over the last 2 or 3 years of the different options in this early relapse setting. And so how do you personally work through all of those options? Joe, maybe you start with you.
And this is the most complicated part of myeloma, isn't it?
Yes.
Is trying to understand. And I think we start by saying no one has the right answer. Not that we don't agree, but there are a lot of great opinions in the matter.
Yep.
I try to look at it and say to patients, we have these 3 major novel classes, as it were: proteasome inhibitors, immunomodulatory drugs, and monoclonal antibodies.
Okay,
and
good.
of course we add steroids or other things potentially to it. But really we've got 3 big players in the field. And I think we've generally learned from a lot of these studies, as the ones you've presented, that using 2 out of them is better than just using 1 in most patients. There may be some exceptions to that rule.
Right.
So then we sit down and say, what have you been treated with before? And secondly, what are you on right now? I'm nervous if Paul has said so clearly that lenalidomide maintenance is the backbone of treatment. If someone is relapsing on lenalidomide, I'm a little nervous to use lenalidomide right then in the next phase. So our usual go-to would be something they've not been on or not currently on, which naturally tends to favor the daratumumab option because we're not routinely using daratumumab up front.
Right.
So if I've had a patient, for example, who is on the classic VRD and a transplant and lenalidomide maintenance, often the first thing I'm looking to is either towards a daratumumab plus a proteasome inhibitor like bortezomib,
Right.
or yes, indeed, perhaps carfilzomib, and sometimes we favor adding pomalidomide to it by virtue of the fact that someone's relapsing.
So you could move from Revlimid to pomalidomide.
Correct. Now, on the other hand, we still have lots of other choices. If someone really desires an all-oral combination, there's the ixazomib options,
is a good option,
whether it's with cyclophosphamide or pomalidomide, or of course lenalidomide if they're not on it currently. So we have lots of choices, but I would start with that basic. Here are the three big players. We tend to want to use two of them. Which two fit better?
Absolutely.
And then also a reassurance to the patient that at some point they're quite likely going to see all of them. So the sequencing, as we know right now, isn't perfect.
Right.
But we want to give them the best depth of response possible with the least amount of toxicity.
So any additional perspectives? We also additionally obviously have the analysis that was done to show that the dominant impact of DARA in this setting. I mean, it does impact your thinking.
Yes, it does. I think the important point, though, is that, at the end of the day, this is certainly not a zero-sum game. And I think the important point for our patients is the beauty in this 1 to 3 prior space is that we have lots of interesting choices.
Yes.
Ones that we can potentially keep in reserve. And I personally extraordinarily impressed with DARA, obviously from beginning to now. But I actually use an antibody called elotuzumab more often perhaps than others do in early relapse, because it's exquisitely well tolerated.
Well, that's obviously the positioning for it.
Exactly, and it's a very well tolerated antibody, and I find it very useful, particularly in these biochemical progressions where you don't have a significant MDE, myeloma-defining events, and you can exploit the opportunity because ELO is, again, the toxicity profile is remarkably favorable. And we do a lot of work with it. My partner, Jacob Laubach's done some beautiful work with it, and my other colleague, Andy Yee, has pioneered a trial looking at elotuzumab, pomalidomide, bortezomib, and dex with the MMRC, and it's been remarkably effective as a salvage platform. And it keeps the DARA in reserve. Not that I'm arguing you have to, but it just gives you that choice. And I think the other thing is that I agree with you, Joe, that in the 1 to 3 prior setting, you obviously have to adapt. So if in my view, if you have an aggressive relapse, particularly there was a nice paper in the poster session looking at outcomes, for example, within 12 months of transplant relapse. That's a particularly high risk group. There, I would absolutely agree with you. A daratumumab-based approach belongs because that's a bad acting group, and that disease can be very, very dangerous. So I think going in with your maximal therapy at that point, and I agree with you, Joe. I like Carpom-Dex. I think it's a great salvage strategy. And then, of course, you've got the DARA options.
You remind me that there was another abstract that I didn't highlight there that was presented, which was pomalidomide Cytoxan Dex, which was used for patients relapsing coming off VRD, which is remarkably active, as a matter of fact.
Exactly, and an all-oral option with the chemotherapeutic to assist in efficacy. Yep.
If I can give a little thought for something that maybe in the future that we're not there yet, something that we're working on aggressively. Keith Stewart and his group and Rafael Fonseca and their group have done some great work in the lab, creating a model that can actually take a sample from a patient's marrow and test it against multiple different drugs that we have, literally every drug against that we have the FDA, and get a sense. I mean, it's still a research purpose. We're still not doing it prime time. I had a patient last week who's been on elotuzumab, lenalidomide, dex, and one of these assays assesses their sensitivity to lenalidomide, because we were trying to see, are they more resistant to len, are they more resistant to ELO? And the patient showed results that were very resistant to lenalidomide. So that might push me in a different direction.
This is very interesting for me to hear. This is deja vu for me, because the human myeloma stem cell assay was developed by Sid Salmon.
Yes, back in
the day.
Arizona.
And what we set up was to test the sensitivity of myeloma to all the different drugs that we had at that time. The problem was that we didn't have that many.
And if you think down the line, I know, again, I don't want to think too Star Trekian, but wouldn't it be ideal if we take someone's disease and say, look, these are actually the best drugs for you?
Right.
Yes.
That's
right.
individualized medicine.
Absolutely.
Let's save you the toxicity and the cost of the ones you don't need.
Absolutely.
Let's use the ones that are most
Which
effective.
was actually the way that this system was set up. And I know that Mikhael has this cool system where it's based on the weight change of the cells, right? Where it goes through this really complicated little tube system.
Yes, but there are multiple different ones. There was a lovely poster who got an award actually from Konstantin Mitsiadi's group looking at a very sophisticated CRISPR-based technology that was really, I mean, the science was, talk about Star Trekian, this was way ahead. And the lead author, Sarah Gandolfi, won an award from ASH as the best, one of the best poster abstracts, where really they've built a platform, to your point, Joe, that will start to identify, you know, particularly in such granularity, therapeutic approaches that might make sense using this particular CRISPR-based platform with gene knockouts and so on to really drill down, to your point, so that we go, you know, from Sid's point of principle, Raphael and Keith's excellent work that, you know, provides a model system, to even deeper granularity that allows us to be that much more precise.
So I think that this could be very cool moving forward.
It could.
Lots of exciting things. Absolutely. So the final segment is very exciting, and I think especially this year, and this new therapy session includes one where I think that it's probably had the biggest buzz of ASH has been in this segment. So I'm going to show you some of what we've heard here in Atlanta and get your personal feedback about some of these results, okay? So I'm going to start with what we've heard about before. I just draw attention to the fact you guys have been involved with these studies using venetoclax, the BCL2 inhibitor, monotherapy, very interesting activity in 1114 related to the BCL2 level, published in Blood just November 30th, and then the combination data combining with bortezomib, activity especially in the bortezomib naive, but also in previous exposure, again just published in Blood. Very, very interesting sort of precision approach. Then the trials are continuing with selinexor, and we have the STORM trial moving forward. I think that a little bit more challenging, I would say personally, related to toxicity, but really, really important to have one more agent that is active in this very refractory quad, penta-refractory situation, so very important. Then we do have the other antibody, the izatuximab, and I'll ask you about this, Joe. This is you. Actually, both of you were part of this. Both of you. So I think we mustn't forget that there is actually another anti-CD38 coming along, and then of course the abstract which probably attracted the biggest buzz at the meeting, which is the anti-BCMA CAR T-cell therapy, the so-called Bluebird trial, the BB2121 there. So I draw attention, a small study, 21 patients, and CRs at 56%, VGPR 89%. So obviously deep responses in very, very heavily pre-treated patients. Up here you see this. But I would also draw attention to this down here. You know, not all of these remissions are being sustained. Certainly there are some MRD levels being achieved, but I think that we've talked about MRD. One needs to look at that carefully. What is the level of the MRD? Is it sustained? And I think balancing that efficacy, what about the toxicity, the cytokine release? Quite challenging. Most of the CRS was grade 1 and 2, but certainly grade 3, got to be very cautious about that. And then the immediately following abstract, abstract 741, was the anti-BCMA monoclonal antibody with the drug conjugate. Again, very, very interesting results. This was an expansion of a study presented last year, actually. 60% response rate, but with deep responses, stringent CR, 15 VGPR, so rather well tolerated, an unusual corneal toxicity, which apparently is quite manageable, but needs to be watched. And so very, very interesting to see these two approaches to the same target. And so finally, I just want to say that we have all of this different information in these different classes, the monoclonal antibodies, the immune modulatory agents, the proteasome inhibitors, and these other classes of drugs. Very, very fortunate to have all of those. And so my question to both of you, maybe Paul, you would like to start first on this one. What do you feel is the most promising approach right now?
Well, you know, I was asked to do a sort of best of ASH for a large patient website, and it
Yes.
was, of course, the CAR-T platform. I thought the results were remarkable that Hez has presented. I think, again, you know, an overall response rate of 94%, a CR rate north of 50%, and the VGPRs, as you showed so nicely. So really impressive. I think the flu-cy platform does appear to matter because Adam Cohen presented a very nice presentation, a very thoughtful presentation from the UPenn group the day before,
Right.
where 4 of 9 patients responded with only Cytoxin. So what this tells you, I think, about CAR-T...
But I think the toxicities were a little challenging in that particular study, as I recall.
Exactly. But what it tells you, and I couldn't agree with you more, Brian, but my point is what it tells you is that this is a highly nuanced approach and that subtle changes may make big differences. So there's a lot to learn. And obviously we have the strength of the Chinese data, which I think is fascinating.
Yes, the Shanghai
and they're
team,
using
yes.
this two antigen approaches, which has been really impressive.
So it's interesting to think of these in terms of both efficacy and toxicity.
Absolutely. And that's my point, is that it's highly nuanced. And I think that CAR-T is fantastic. I mean, it's clearly one of the most interesting and promising new approaches. But I think we have to be careful because it's early, and it's essentially, when you look at these experiences, they're essentially a numerator-based experience because we were part of the Bluebird trial, and you have, you know, a waiting list that's almost a year long to get onto these studies.
Right.
So there's selection in that process.
Absolutely.
Irrespective of that, the data are very, very impressive. I think the CRS is relevant. We have to remember in myeloma patients, because the allo experience tells us this, our patients are vulnerable to immunotherapeutic toxicity. And the best example of that is what happened with checkpoint inhibition and IMiDs in the Pembro studies.
Yes,
so
yes.
we have to be very aware of you know, great efficacy, but let's be also careful about side effects.
Right. So
and you know, that's
that's
relevant.
very, very important. And so Joe, what's your take on this? The CAR-T and then the antibody?
So much to say, but very briefly, you know, I think those first few molecules you noted, the venetoclax,
Yes.
you know, it's, if you will, a boutique drug to some degree, at least now by
But very
itself.
helpful, very helpful.
We've been able to use it off study in a number of patients with translocation level 14. It's a pill, very well tolerated. Some of our patients with very aggressive disease just seem to immediately respond to this drug. Selinexor, I agree with you. It has tremendous challenges from a GI toxicity, but if we can get people through that first month, often it's much more tolerable. I do think isatuximab will have a role in you know, I think the more the merrier in some respect. That's right, we work on it together. We have to. You know,
the PI
daratumumab has been so prolific, it won't have that same effect, of course. And now we're working with an even more rapid infusion of the drug, where it can be given over 75 minutes.
Right.
My quick take on the CAR T-cell therapy is very similar to what Paul has said. We're all excited about it.
Yes.
But you know, we're still got some early model CARs here. I mean, we really need to work out a lot of the bugs. Some of these are a little bit clunky. They're not quite efficient yet. I don't say that to be disparaging. I just mean that not all CAR T-cell therapy is the same. And we learned from our leukemia and lymphoma colleagues, it took them a number of years to refine this. And although, as you say, waiting lists are long, I think the CAR T-cell therapy that we're going to have at 1 year and 2 years from now will be that much better, where it will be more efficacious and it'll be safer. I am a little nervous at the number of people that have relapsed fairly soon thereafter. So this idea that people think it is a cure, or that people think it's just a be-all and end-all, we have to be cautious. It's like any other treatment we've had, albeit very promising in very relapsed disease. So,
this last point about where patients are relapsing, and I would balance that against what will undoubtedly be an enormous cost. And so with the approval of the CAR-T in the childhood leukemia setting, this raised that issue, and the number that started to float around immediately was several $100,000. You know, between that and $1 million, once you consider all the logistics and you reach a high commercial level of this preparation, which is by no means trivial.
And that speaks to access too.
Yes.
So it's not just the pure cost, it's how many people are going to have it access. I mean, looking way down the line, we'd love an off-the-shelf like CAR T-cell approach where it doesn't have to go through all the preparation, but we're clearly not there
No,
yet.
that's a little disappointing, actually, initially, right?
Very much so. So time will tell.
Yeah, and if I may, I'll pick up on that point, Joe, because I think it's an excellent one you make, the question of being off the shelf. And that's what I really like about the BCMA antibody drug conjugate study that Suzanne presented. And again, in full transparency, I'm obviously a co-investigator there. But the fact is, what we saw with that antibody, every 3-week infusion, you know, it's a 1 to 2 hour infusion. It's a walk in the park by comparison. The corneal toxicity, to your point, Brian, is very manageable.
Yes.
You've got to have a good ophthalmologist, but it's straightforward. We saw thrombocytopenia, but really beyond that, not much else. And I think that kind of striking efficacy from a monotherapy, and it's worth noting in Suzanne's presentation that responses were seen in 40% of patients who'd had prior DARA. And I think that points to the success of the future. And one last closing comment, I completely agree with you, Joe, that CAR T is great, but what we mustn't do is think of this as a sort of zero-sum game. And there were some very nice presentations of additional drugs where we're seeing salvage in the setting of relapse refractory disease. And a variety, you touch on the selinexor, which I think is absolutely correct. Fascinatingly, a straightforward modified alkylator called melflufen. Mary V presented lovely data in the Horizon trial.
So a lot more to learn in this setting. I would have to say I found the results with the GSK monoclonal antibody intriguing. Deep responses, well tolerated.
I agree.
And so I think that we need to continue, watch this space. Watch this space and see what emerges. But really tremendous hope and excitement. Hope for all patients who might be watching. A lot in the pipeline, a lot very close to being available. So thank you to you both for really offering your wisdom and insight to all of these aspects of myeloma. So thank you to all of those who have been watching and listening. Hopefully you found it to be worthwhile. Thanks again to our speakers.
Pleasure. That's a pleasure.
Thank you.